Tumor, and today I will be talking to you about the isolation and molecular profiling of these CTCs in head and neck cancer. My mentor is Dr. Jonathan Beitler, and Dr. Xu Wang helped me with all the laboratory techniques. Here's a schematic diagram of what happens in the human body. You have a primary tumor, and some cells detach from this tumor via what is thought to be EMT, epithelial to mesenchymal transition. These cells have the potential to travel in your blood or circulate and then travel to other parts of the body and start new tumors there. So my research task and overview, I took a tissue specimen from the patient and I used QD for IHF, which is quantum dot immunofluorescence staining, to study the molecular profiling
and any possible biological differences between cells of the primary tumor and cells in the blood. Both are cancer cells. Also, I studied a technique called isolation by size of epithelial tumor cells, or ISET for short, where I used a filter to separate cancer cells from normal cells with a syringe pump. So two parts of my study were first to optimize the conditions for filtering to get the best capture efficiency or highest number of cancer cells retained on the filter membrane after filtering, and also molecular profiling to validate the use of nanoparticles. The nanoparticles I used were quantum dots, but more advanced nanoparticles are gold nanoparticles, which is part of a bigger project. So first, before I started filtering, I had to think about which size filter I should use. There are various sizes, so 5, 8, or 10. So first I had to study the average cancer cell size. So lots of tissue samples on various slides. I stained these with eosin, and I did not dehydrate them because I found that dehydration actually shrinks the cells, thus causing a dramatic change in cell size. I had 9 cancer cell slides, all head and neck cancer, and also 4 controls. You can see from the images I took on the microscope that cancer cells are visibly larger than normal cells. Yes? Then I created a bar graph with average cancer cell size. So the blue bars represent cancer cells, and the last 4 bars represent normal cell lines. From this graph, you can see that the cutoff for cancer cell lines is about 10 microns. But actually, the filter I chose had 8 micron pores, which means that any cells that are bigger than 8 microns will stay on the filter, while cells that are smaller or 8 microns will pass through the pores. So that's why I wrote the question, why did I decide to use the 10 microns instead of the 8 one? So then I started filtering with this 8 micron size filter. It's a polycarbonate filter membrane manufactured by Isoport. I used a syringe pump and varied the filtration speed on that. The filtration speeds I used were 0.1, 0.2, all the way to 0.5 milliliters per minute. I conducted 3 independent experiments for 2 head and neck cell lines, TU212 and JHU012. Here are the 2 graphs for the TU212 and JHU012. For the first graph, you can see that filtration speed had more of an impact on the percentage of cancer cells that were retained on the membrane. But if you look at the other one, you can see that the results are more linear, which means that changing the filtration speed did not have much an effect. That means that the filtration speed should actually be varied for each cell line to optimize the recovery rate. Next, I used the TU686 GFP, which means green fluorescent protein cells, which are cancer cells. And I spiked these into 2 milliliters of mouse blood, which contain normal white blood cells. So my purpose was to see in a real situation, for example, modeling patient blood sample, how many cancer cells can you recover in a mix of normal and cancer cells. So I put the flow rate at 0.3 milliliters per minute and used the 8 micron pore size filter. In one trial, I found that I could recover 4 out of the 10 cancer cells that I had spiked into this mouse blood. The FITC shows the cancer cells, which are really big and really bright. because of the green fluorescent protein. And the DAPI staining was used to show the presence of the nucleus to confirm if they were cells or some other foreign particles. And the viable cells were extracted from mouse fluid. My next part of the study was to use quantum dots, which are a type of nanoparticle, to study any possible changes between CTCs, which are in the blood, and cells of the primary tumor.